So hello everyone, my name is Debprosit and I'm a currently second year MBA student and I'm from AIMS Bhopal. So today's topic will be from Unit 9, that is Biotechnology, the Chapter 1, that is Biotechnology Principles and Processes. So, just a general introduction of the chapter. So, in this chapter, we discuss the principles and the processes of biotechnology. What is biotechnology? In biotechnology, we actually deal with recombinant DNA technology, where we genetically modify a DNA and then genetically modify an organism, and then we get the product that we want. For example, insulin. So earlier what used to happen was we used to take insulin from cattle, and then we used to inject it into humans, and that had a lot of problems, like the insulins from cattle was not actually for humans. So many people developed reactions against it, and also it was not as much effective, and also the amount the amount of insulin was very expensive back in the days, because the amount of insulin that was being produced was very less. But with the evolution of biotechnology and with the evolution of recombinant DNA technology, we were able to collect the insulin gene from our human body, and then we were able to introduce it into E. coli, and we were able to produce insulin in a mass number, and now insulin is quite cheap and easily accessible to everyone, and it is actually the human insulin. So the chances of developing any allergic reaction to it is also very less. So this is biotechnology, where we genetically modify an organism in order to create a product, the desired product that we want, and then after getting the product, we process it. It goes through a downstream processing, and after that it is launched in the market. So this chapter basically deals with all the principles that are used in it. For example, PCR and restriction enzymes, and all those terms you'll learn in this chapter once you read it. So let's start with the exercise. First question was, can you list 10 recombinant proteins which are used in medical practice? Find out where they are used as therapeutics. And so I have listed, I've made a list about 10 recombinant proteins. So first one is insulin, as I said, and it is used in treatment of type 1 diabetes mellitus. And second one is interferon alpha, used for chronic hepatitis C. Interferon beta, used for herpes. Factor 8, used for hemophilia treatment. In hemophilia A patients, the factor 8 is deficient. That's why we give them factor 8 artificially. And factor 9 for hemophilia B patients, and DNase 1 enzyme for cystic fibrosis treatment, and antithrombin 2 for preventing blood clot, and interferon B. So interferon B and interferon beta, these two are different. Beta is used for treatment of herpes, and interferon B is used for treatment of multiple sclerosis. And human recombinant growth hormone, it is used for promoting growth, and tissue plasminogen activator, used for acute myocardial infarction treatment, which is basically in treatment of heart attack. So these were the list of 10 proteins that are commonly used as a recombinant protein in clinical practice. Now question number 2, make a chart with diagrammatic representation showing a restriction enzyme, the substrate DNA on which it acts, and the site at which it cuts the DNA, and the product it produces. So I have taken the example of EcoR1, as it is in our syllabus. So it acts on GAATTC. This is a palindromic sequence of DNA. So it acts in this sequence, and it cuts between G and A, and it produces DNA fragments with sticky ends. So for better visualization, here's the diagram. As you can see, this is the host DNA, and this is the vector DNA. So what happens is EcoR1 comes and cleaves the DNA between G and A, GNA and GNA, and after that we get sticky ends like this, and these sticky ends come close to each other, and then they form bond and they are joined by ligase enzyme. So that's how the process works. Now coming to the next question, question number 3. What from what have you learned? Can you tell whether enzymes are bigger or DNA is bigger in molecular size, and how did you know? So the answer is DNA is bigger. An explanation. So explanation is structure of DNA is more complex than enzymes. For example, DNA has nitrogen base, pentose sugars, and phosphorus moiety, whereas enzymes only has amino acids that are bound with polypeptide chain. So structure of DNA is more complex. So this is one point. And the second point is you can think of it like genes code for enzyme, and genes are just a fragment of DNA. The entire DNA is much bigger than this small fragment that is coding for an enzyme. So enzymes are coded from a fragment of DNA. That's why enzymes must be smaller and the entire DNA must be bigger. So this is a common sense. What is our answer? Based on this, DNA is bigger in molecular size. Now coming to question number 4. What would be the molar concentration of human DNA in a human cell? Consult your teacher. So this is actually a question that involves the concepts from physical chemistry. So concentration, I have taken molarity, and as we know, the definition of molarity is number of moles of solute. In this case, the solute is DNA, are present in unit volume of the solution. In this case, we are taking the volume of cell. So this is the basic formula of molarity. Now let us take a single base pair. The molecular weight of a single base pair is 650 Dalton. And how many pairs do we have in our genome? We have 3.3 into 10 raised to the power 9 base pairs. So the total weight of DNA is total number of base pairs multiplied by the molecular weight of a single base pair. So we have the total weight of DNA that is present in our genome. And we also have the molecular weight of a single base pair. So from this, we can easily calculate the moles. What is mole? Mole is total weight divided by molecular weight. So number of moles is 3.3 into 10 to the power 9 billion. Sorry, 3.3 into 10 to the power 9 into 650. This is the total weight divided by 650. This is the molecular weight. And this will cross out, and the volume of cell. So for a volume of cell is approximately 10 to the power minus 12 liter. This is just a data you have to memorize. So the answer that we are getting is this 3.3 into 10 to the power 9, which is the number of moles of base pair, and 10 to the power minus 12, this is the volume of cell. And the final answer would be 3.3 into 10 to the power 21 molar. And we can write it as the molar concentration of DNA is 3.3 into 10 to the power 21 moles of base pair per liter. So notice this, I have said moles of base pair. Okay. We are calculating the molar concentration of DNA on the basis of moles of base pair. Now, question number 5. Do eukaryotic cells have restriction endonucleases, and justify your answer? So the answer is no. Eukaryotic cells does not have restriction endonuclease, and the reason is eukaryotic DNA are generally protected by methylation. Methylation is a process in which the enzyme methylase adds a CH3 group to the DNA, and what it does, it protects the DNA from restriction endonuclease and several other enzymes also. And for this simple reason, eukaryotic cells does not have restriction into nuclease enzyme in it because the eukaryotic DNA is already protected from RE. Now, coming to the next question, besides better aeration and mixing properties, what other advantages do stirred tank bioreactors have over shake flasks? So we use shake flasks generally in laboratories just to see if the GMO or genetically modified organism is producing the protein or not. And we use the bioreactor when we have to produce the product, that is the protein, at a large scale for industrial level. So we use bioreactor when we have to produce the product at a large scale, because we have to make the process efficient for that. So what are the advantages of bioreactors over shake flask? That is the question. So the first would be for sampling and testing of a small volume of culture can be taken out of the reactor. So when we use stirred tank bioreactors, we can easily take out a small sample of the product, and then we can test the product. We can test the quality of the product while it, and then if we find something that is missing, something that is not right, we can then change the conditions according to that, and we can then modify the conditions, and we can then create the optimum conditions to fix that. And we cannot do that in shake flasks, but we can do that in bioreactors. So this is one advantage. Second advantage is it has foam breakers for regulating the foam. So it does not, it doesn't let foam to generate, and that's why it increases the aeration. It increases the, breaks the foam, so it increases the surface area, and it increases the aeration, and it makes the process much more efficient. And the third point is we can control pH and temperature in stirred tank bioreactors, but we cannot control it in a shake flask. So these are the three advantages besides aeration and mixing properties that bioreactors have over shake flask. Now coming to the next question, question number 7. Collect 5 examples of palindromic DNA sequence by consulting your teacher. Better try to create palindromic sequence by following the base pair rules. So I have created 5 sequence here. As you can see, GATTC, GAATTC. So this is a palindrome. Also the next example, GATATC, GATATC. So and also the next example, I'm going from 5' to 3', so I'm getting CAG, CTG. Also from going to 5' to 3' in the complementary strand, we can get CAG, CTG. So this is creating a palindrome. Here are the 5 examples. Next example, just see that. So how do you easily create that? So there's a very simple method of this. First, write any 3 base that you want. For example, I'm writing ATC. Randomly select any 3 base, okay, and just repeat these 3 base here, ATC, okay, and now just put the complementary bases over this. So I have to put T, I have to put A, and I have to put G over here, and I have to put G here, A here, and T here, and give it polarity 5' to 3', 5' to 3', and you have a palindrome. So what we did, we just selected 3 random base ATC, and then wrote it here, and then just filled the rest part of the DNA, and we have a palindrome that is ATC TAG. And sorry, I made a mistake. You have to write it in the opposite sequence. ATC. So you have to write it like ATC. I made a mistake of writing it like ATC and ATC. Not like that. You have to write it like this. ATC and ATC. Okay. So this is going from 5 prime to 3 prime. This is going from 5 prime to 3 prime, and you can just now use the rule of complementary to fill the rest of the bases A and T and G, A and T. And now we have the palindromic sequence, that is ATC, GAT, ATC, GAT, and that is the same from the both sides. So this is a simple method to create a palindrome. And now question number 8. Question number 8, can you recall meiosis and indicate at what stage a recombinant DNA is made? So meiosis, if you recall, it has two stages, meiosis 1 and then meiosis 2. Meiosis 1 has prophase 1, metaphase 1, anaphase 1, and telophase 1. And in prophase 1, if you recall, there's a stage called pachytene. And in pachytene, the crossing over occurs. And what is crossing over? Crossing over is essentially recombination, where a part of non-sister chromatid is inserted into another non-sister chromatid. So it is basically recombination, where part from a segment from a different segment from a DNA is getting inserted into a different DNA. And this stage is pachytene. So answer would be pachytene. Question number 9. Can you think and answer how a reporter enzyme can be used to monitor transformation of host cells by foreign DNA and in addition to selectable markers? So what is a reporter enzyme? Reporter enzyme is actually the enzyme that is created by the selectable marker. So let's take an example. I want to create a genetically modified organism. So for that, we have to take a gene of interest. So this is the gene of interest, and this is the vector. And it has a selectable marker, that is LAC-Z gene. This is the selectable marker. And it produces the enzyme beta-galactosidase, which is the reporter enzyme. So what happens, I'm using RE and RE to introduce the gene of interest into the vector. So there will be some DNA that will be recombinant, and recombinant DNA will get inserted into the site of LAC-Z gene, and it will cause insertional inactivation. So beta-galactosidase enzyme, that is essentially the reporter enzyme, it won't be produced, and in non-recombinant DNA, the enzyme would be produced. So what happens is there are 3 types of cells that can be that we can get in the solution. One is non-transformant, another 2 are transformants. And in transformant also, we get non-recombinant types and recombinant types. So in non-transformants, the plasmid is not introduced. So these are non-transformants. So in the X-gal medium, they won't produce any color because they are not producing any enzyme. So they won't produce any color. And the transformants, but non-recombinants, will produce beta-galactosidase enzyme that will degrade this X-gal to form a color that is green. So they will show green fluorescence. The non-recombinant transformants will show green fluorescence. And the recombinant transformants will produce a different enzyme because of this insertional inactivation. Okay, and it will produce a blue color. And that's how the reporter enzyme is used in RDT to differentiate between the recombinant transformants, non-recombinant transformants, and non-transformants. Hope you got that. So here, this is a selectable marker produces a reporter enzyme, and we can use this reporter enzyme to differentiate between transformants and non-transformants. So this is the basic principle. Now, question number 10. Describe briefly the following. First one is origin of replication. So origin of replication is a particular site at which the replication process starts. So if you take, this is a plasmid, double-stranded circular DNA, or we can just say plasmid. So, and you can see this point, this is origin of replication, which is also which can also be denoted as ORI, ORI. This is essential part of the DNA in order for the replication process to occur. Without ORI, the replication process will not occur, and the gene will not get replicated, and the gene will not get transferred to the next generation. So for replication process to occur, ORI is very, very important. And we have to keep this in mind while using a plasmid for recombinant DNA technology, because if we use a plasmid and it does not have an ORI, I mean, for some reason, suppose the plasmid does not have an ORI, but we're using it. And even after inserting such plasmid into the bacteria, we won't be getting any protein from it, because the plasmid won't undergo any replication process inside the cell. And if it doesn't undergo any replication process inside the cell, it won't produce any protein, and we won't get any product. So entire process will be wasteful. So we have to keep in mind that ORI must be present in the vector in order for a successful RDT process. So this was origin of replication. Now coming to the second, bioreactors. So what are bioreactors? Bioreactors are actually an engineered system that can be used to grow microbes at a large scale. And NCERT mentions two kinds of bioreactors. So this is the simple stirred tank, and this is the sparge stirred tank. So what happens in sparge stirred tank? We create bubbles. We create bubbles like that. So when the bubbles go up, they come in contact with the surface and they get burst. And by this process, they create unevenness on the surface. And what it does, it increases the surface area. And by increasing the surface area, it increases the area for oxygen transfer. And this increases the efficiency. So this is the only difference between sparge stirred tank and simple stirred tank. In simple stirred tank, there are no bubbles to maintain its homogeneity. But in sparge stirred tank, along with maintaining homogeneity, it creates bubble that increases surface area for gaseous exchange, so thus making it more efficient. So this is bioreactor, and you can just read it. What are bioreactors? And these are generally used to produce the products at large volumes in hundreds or thousands of liters, and that is it. Bioreactor provides the optimal conditions for achieving desired product by providing optimum growth conditions. You can change the temperature, pH. You can control the temperature, pH, substrate, salts, vitamins, and oxygen throughout the process. You can constantly monitor the process, and you can constantly make changes so that the process is efficient and cost effective. And the third was downstream processing. So what is downstream processing? Downstream processing is the series of processes that a biosynthetic product undergoes before it gets launched into the market. So basically, two processes that occurs. One is separation, and second is purification. So in a bioreactor, we are creating the product. So this is the solution. In this solution, the product and the bacteria, everything is mixed. So what we have to do, we have to separate the product from this mixture. So the first process is separation. And after separating, we have to purify the product. We only need the product in our final product. I mean, we only need the protein that we want in our final product. We do not want anything else that is already mixed in it. So we have to purify. So for that we use several enzymes and preservatives and things like that. So these two processes are collectively known as downstream processing. You can also read here. The process include separation, purification, which are collectively referred to as downstream processing. And the product has to be formulated with suitable preservatives. Such formulations has to undergo thorough clinical trials in case of drugs. So before launching any drug to the market, we have to do clinical trials of it, which is a common sense. So this whole process is known as downstream processing. Now, question number 11, that is PCR. It is explained here also. So PCR is polymerase chain reaction. So what we do is basically we extract DNA from a sample and then amplify it in order to make it noticeable or measurable, and we can use it as diagnostic tests. So the basic procedure that PCR follows is first denaturation, second is annealing, and third is extension. So in denaturation, we increase the heat up to 94 degrees Celsius. So the two strands of DNA get separated. And then in annealing, we add a primer, RNA primer. We reduce the temperature to 54 degrees Celsius now. And then we increase the temperature to 72 degrees Celsius again. And the primer is attached. Now the DNA polymerase will come and cause extension and will form new DNA strand. And by this process, we are creating two DNAs from a single DNA. And after repeating this cycle several times, we can exponentially increase the amount of DNA that is present in solution. Okay, so this is the use of PCR. This is the basic principle of PCR: denaturation, annealing, and extension. And we use PCR when the DNA that is present is very less. So we have to use PCR to make it to increase the number of DNA, so that we can perform as many tests as we want. Okay, and the DNA polymerase that we use in this PCR, we extract it from a bacteria, Thermus aquaticus. This DNA polymerase can act at high temperatures. So we use this one. Now the second was restriction enzymes and DNA. So restriction enzyme, I'm just going to refer to NCERT for this. So NCERT has excellent description of it. So as you can see, here's the history in year 1963. The E. coli, this RE was isolated from E. coli and everything else. So and which was later called as restriction endonuclease. So the first restriction endonuclease that we got was HIND2, which was extracted from Haemophilus influenza. Okay, so this was our first RE. It was found that RE always cuts the DNA molecule at particular point by recognizing a specific sequence of 6 base pairs. So this is known as recognition sequence. So what an RE does, RE finds a recognition sequence that is generally a palindrome, and then cleaves the DNA at the recognition sequence. Okay. And now we have more than 900 restriction enzymes that are have been isolated from over 230 strains of bacteria. So this was just an information. And how do we naming of RE? This is very important. So the first letter of the name represents the genus. As you can see, H, I, 2. H represents the genus Haemophilus. H represents the genus. And the second two letters come from the species. IN, it denotes influenza. And the fourth letter, that is D, it represents the strain. In case of Eco R1, it is Escherichia coli, and the strain is RY13. So R basically represents the strain RY13 in case of Eco R1, and the number, the Roman number, it represents the order in which the enzyme was isolated from the bacteria. So R is derived from the name of the strain, and the Roman numbers following the names indicate the order in which the enzyme were isolated from the strain of bacteria. And restriction endonuclease, as you can make from the name endonuclease, it has endonuclease in its name. So restriction endonuclease are a branch of they belong to a larger class of enzyme that are called nucleases. And in nucleases, there are two types of nucleases, exonucleases and endonucleases. So restriction endonucleases are basically examples of endonuclease enzymes. So these cleave the DNA at any part other than the ends. Exonucleases cleaves the DNA from the end, and endonucleases cleaves the DNA at any part other than the ends. So this is the basic description about restriction endonuclease that is important for NEET. And also, as I said, the palindromic nucleotide sequence. This is the recognition sequence that an RE recognizes. And number 3 is chitinase. Chitinase is basically an enzyme. It dissolves chitin that is present in the fungal cell wall. So what is the use of it? You can see it is written here. So when we have to isolate the genetic material from a fungus, DNA is actually enclosed within the membrane, and so that's why we have to break the cell open to release the DNA along with other macromolecules such as RNA. And for that, we have to break the cell wall of fungi. And the cell wall of fungi has chitin in it. And that's why we are using chitinase enzyme to get the DNA that we want from that fungus. So this is chitinase and nothing else. And coming to the last question, we have we have 3 differences. So the first is difference between plasmid DNA and chromosomal DNA. So I have written it here. Chromosomal DNA, as you can see, chromosomal DNA is the genomic DNA, and plasmid DNA is the extra chromosomal DNA. So for example, this is E. coli. So this is the main genomic DNA of E. coli, that is double-stranded circular. And this is this small part that I'm making right now. This is plasmid. This is extra chromosomal. And as you can see, the chromosomal DNA is larger and plasmid is smaller. And chromosomal DNA is actually found in both prokaryote and eukaryote, and without chromosomal DNA, the survival of the cell is impossible. So we need chromosomal DNA in order to survive. But plasmid is found only and only in prokaryotes, and a cell can survive without plasmid also. I mean, a cell does not need plasmid for its essential functions. Plasmid gives the cell additional functions like protection against several antibiotics. but it is not an essential function like metabolism or anything like that. So SL can very well survive without plasmid also. And chromosomal DNA undergoes vertical transfer and plasmid undergoes horizontal transfer. So this is basic. Vertical transfer is basically cell division. So when a cell divides, it divides into two, and it transfers its genetic material into these two cells. So this is known as vertical transfer. But the transfer of plasmid occurs by horizontal transfer, that is transduction. So what happens is this is a bacteria, and this is the second bacteria. So this one has plasmid. So what happens is a pilus known as sex pilus grows from this bacteria, and then it integrates with the another bacteria, and plasmid through this pilus gets transferred into the second bacteria. And that's how the transfer of plasmid goes. I mean, transfer of plasmid is generally in the same generation. That's why it is known as horizontal transfer. But the transfer of chromosomal DNA is from one generation to the next generation. So it is vertical transfer. Now, second is RNA and DNA. RNA is single-stranded and DNA is generally double-stranded. RNA is more active, as we also know that RNA can act as enzyme. So enzymes are actually enzymes are generally active. So RNA is more active and DNA is less active. And it is owing to the fact that DNA is double-stranded. So due to the presence of more stronger bond in DNA, it is less active and more stable. Okay, and RNA has uracil and DNA has thymine, and other bases are same. So this is the point of difference between RNA and DNA. And also RNA is less preferred as genetic material since it is more active. So more chances of mutation are in RNA, but DNA are more stable. So these are more preferred as a genetic material than compared to RNA. So these are the basic difference between RNA and DNA. And coming to our last question, that is the difference between exonuclease and ended earlier that it cleaves DNA at places. Sorry, I made a mistake here. Endo, and this is exo. So endonucleases cleaves the DNA at places other than the two ends. It can cleave the DNA anywhere other than these two. Anywhere other than these two ends. An exonuclease cleaves the polynucleotide chain by removing nucleotides one by one at the ends. So this position and this position, these two are the acting sites of exonuclease, and this entire DNA is the acting site of endonuclease. And as we can say that exonuclease acts at the end, so the product of cleavage would be a single nucleotide. So the products are generally single nucleotide. And since endonuclease acts at the middle, as you can say, I mean, it's not end, right? So it creates two fragments of DNA or two fragments of polynucleotide. So the products of endonuclease cleavage are generally polynucleotides or oligonucleotides and not single nucleotide. Examples of endonucleotide include restriction endonuclease, and exonucleotides is DNA polymerase 3. And that's it. This is the basic difference between endonuclease and exonuclease. So this is an